My name is Martin Wiedmann. I'm a professor in the Department of Food Science at Cornell University. Listeria is a group of organisms and includes one which we're particularly concerned about. It's Listeria monocytogenes. And Listeria monocytogenes causes a very severe foodborne illness in humans. And when I say severe, it means about 20% of people who get it die. What happened over the last 5-6 years really is that we and others discovered other Listeria species which from a taxonomy look like Listeria species, but they start to behave differently from Listeria monocytogenes. Most of the testing by industry is really done for Listeria species, and they use the presence of Listeria species as what they call an index organism. And meaning if we find Listeria species, there are probably conditions that could mean we have Listeria monocytogenes there, and therefore we have to intervene. We have to do something to control those conditions to reduce the risk that we ever have Listeria monocytogenes there. So the impetus for this study you referred to was that we found these other Listeria species that didn't quite behave like Listeria monocytogenes, that didn't quite behave like the traditional Listeria species. And so we needed to understand what they are, how do they behave, how do they relate to Listeria monocytogenes, to see whether we actually need tests that detect these organisms or whether the current tests are okay in terms of that they either detect them or they don't detect them. So we wanted to characterize them, to define them. And what we basically found is that many of these species are new Listeria species, and they're very different from Listeria monocytogenes. Some of them don't even grow well at low temperatures, some of them are not motile, don't move around the way Listeria monocytogenes are. So really detection of these species doesn't necessarily indicate that there's an issue with regard to Listeria monocytogenes. So it suggests that we need to readjust and rethink about which species we want to detect as an indicator that there are conditions where Listeria monocytogenes might grow. And the genomics tools are really what will allow us to define pathogens. So it's not going to be yes or no, but it's groups within a species. Some of them are a severe pathogens, some of them are cause less severe disease, and we might have to adjust the regulations and our management practices to be what I call risk-based. We use the risk-based management strategies on the level of which food is a high risk, but we also need to look at the specific organisms. So my goal is really to study all types of bacteria to ultimately help feed people with safe food, but also make sure we can make enough food with preserving the environment, because that's one of the major challenges we have over the next 20-30 years.